Target essay 1. Explain the fine structure of mitochondria. So when I read these, when I describe these things, not in great detail, but when I just point out the essential points, according to the rhythm in which I point out these things, if you can get hold of that rhythm, that pattern, and remember that in your heads in a different way, you all can remember this is better. So explain the fine structure of mitochondria. So you have to draw a fully labeled diagram to get all the marks. Now you see they have given you 10 marks for this diagram. If you don't label anything at all, you don't get marks at all, even though you draw the picture, the diagram of the mitochondria. Essentially, you should name inner membrane of mitochondria. What else should you label? You should label the outer membrane, the DNA. Actually, it should be circular DNA. It's better to be more specific. So inner membrane, outer membrane, and you should specifically say circular DNA. It's better. And then you should show the granules. The granules, the ribosomes, cristae, intermembranous space, matrix, ATP synthase particles. I don't think that's that essential, but you have to label. It's better to label all this. So if you label all this, you may get 10 marks. You will get 10 marks if you really draw a good diagram and label all the components. You can get all the marks. So it is one of the most common organelles in eukaryotic cells. It's one of the most common organelles in eukaryotic cells. So it's found in eukaryotic cells, and it's one of the most common organelles. And it is elongated, as you saw earlier in the figure. It is elongated, stretched. Organelle with two enclosing membranes. You saw how that organelle was enclosed with two membranes, the inner membrane and the outer membrane, with two enclosing membranes. The outer membrane is smooth, you see. So the outer membrane, it is smooth, but the inner membrane is convoluted. Why is it convoluted? Why does it have this kind of characteristic shape in inside one? Outer one is smooth, but why does the inner one has a convoluted shape, irregular shape like that? Because it's convoluted to form what we call cristae. Now, why do we have cristae? Cristae increase the surface area. Now, what do they contain? They contain Stokes particles. The gap or the space in between the inner and the outer membranes of the mitochondrion is called the intermembrane space. The innermost part of the organelle is known as the mitochondrial matrix. So the innermost part of the organelle is called the mitochondrial matrix. Now what does this contain? It consists of 70S ribosomes. Number one. What else? It consists of circular DNA molecule, as I said, circular, or you can say mitochondrial DNA. So number two. Number three, what do you have? Phosphate granules. Number three. Number four, you have enzymes. The matrix carries enzymes. Why do we have these enzymes? For the reactions in the Krebs cycle. Enzymes in the matrix for the reactions in the Krebs cycle, that is in cellular respiration. Further, cristae is composed of proteins. So the further cristae is composed of proteins and enzymes. Why do you need these proteins? Essential. It's essential for what? For electron transport chain and oxidative phosphorylation. So further, cristae is composed of proteins and enzymes, essential for electron transport chain and oxidative phosphorylation. Essay number 2. Describe the primary structure of a transverse section of a dicotyledonous plant stem. Now when you go to the exam, there's a point that you should do. Underline primary structure. You may forget because you're in a hurry, right? You may see it as the secondary structure or something. Of what? Of the transverse section of dicotyledonous plant stem. Dicotyledonous plant stem. Of those plant stems only, you have to describe the primary structure. Now, the outermost epidermal cell layer protects the inner parts from desiccation and infections. The outermost epidermal cell layer. The outermost epidermal cell layer. What does it do? It protects the inner parts from what? From desiccation and infection. The epidermis is interrupted by pores. So what are these pores? They are stomata. The epidermis is interrupted by pores called stomata. Interior to the epidermis is cortex. So what is inside of the epidermis is the cortex. So what does it mostly contain? It contains parenchyma cells. It mostly contains parenchyma cells. So we have to focus on these words. Mostly contains parenchyma cells. The collenchyma cells may also be present just beneath the epidermis. So you may find collenchyma cells just beneath the epidermis. So focus on that. Just beneath the epidermis, you may find collenchyma cells. So where do you find sclerenchyma cells? Sclerenchyma, such as fibers, are also present in the cortex. So in the cortex, we can also find sclerenchyma, such as fibers. Now vascular bundles here are arranged as a ring. So you have to remember that. Vascular bundles are arranged as a ring in dicotyledonous plant stems. In dicot plant stems, you find vascular bundles arranged as a ring. So remember that. You have to remember that. It's a must. It's like this. Around, they arranged like a ring. And you find the pith here. Pith. So I don't describe the whole thing. We go to the image and the main image that they have given us in the marking scheme. We'll go to that and see a clear diagram of it. Now, the vascular bundles are arranged as a ring. The vascular bundle consists or contains, now what does it contain? It contains the primary phloem. Now see, now we are talking about the primary structure of the transverse section of a dicot plant stem. That is why they mention, say this, primary phloem. Vascular bundle consists of primary phloem towards the cortex. So it's pointed towards the cortex. Primary xylem is towards the pith. So primary xylem is pointed towards the pith. Use these words only. Primary xylem is towards the pith. And what do you find in between them? And in between, we have the cambium tissue. In between the primary xylem and the primary phloem, what do you find? We find the cambium tissue. Now, what do you find outside of the vascular bundle? There is a cluster of sclerenchyma cells. Outside of the vascular bundle, there's a cluster of sclerenchyma cells. Now, inner to the vascular bundles, what do you find? There's a large pith inner to the vascular bundles. Now outside the vascular bundles, we saw that there is a cluster of sclerenchymal cells. Now inner to the vascular bundles, there's a large pith, now which is also made up of parenchymal cells. Remember that it's also made up of parenchymal cells. That's okay. So now let's examine clearly the diagram. The diagram is the key. By looking at the diagram, if you can really draw the diagram properly, you can really point out your points while remembering the diagram. So it's better to draw the diagram first, because then you can remember. It depends on the person writing this, but that's a good idea. Now connect this to the other video. We have to discuss the structure. We have to see the structure clearly of the dicot plant stems transverse section. Now we have to clearly label the vascular bundle. The vascular bundle has to be labeled very clearly. That's the vascular bundle. Then we have, facing the epidermis, what do we have? Sclerenchyma, fiber cells. Then we have phloem. And behind phloem, we have xylem facing the pith. We have the phloem facing the cortex, right? So this is the cortex. You can see the cortex region. Then the ground tissue connecting pith to the cortex. So this is the ground tissue that connects the pith to the cortex. And then we have the pith region in the middle. Then we have the epidermis surrounding. So now we'll shift into the essay that's coming out.